Hello. The primordial replicon must have been based on nucleic acids, be it RNA or DNA. Mostly it is likely to be that of RNA is the first molecule, a first propagating entity, and then from which DNA must have evolved by getting rid of the 2' OH so that it has increased stability. This is fine because we understand how the information can nucleic acids from DNA to RNA, RNA to DNA, during transcription too from DNA to RNA. Forming of those is purely based on base pairing, and that is easy to envisage. But what is astonishing, or what is still incredible, is how the sequence of nucleotides can dictate a sequence of amino acids, that is proteins. And that makes translation one of the most intriguing processes, at least in terms of how did evolution of translation happen. That's an incredible one to see. And we call it translation, and say, for example, transcription. It is production of RNA based on the template of as DNA as the template. So it is still in the language of nucleic acids, just in DNA and RNA, so we call it transcription. We call the formation of proteins from the information provided by the DNA as translation, because it is now what was in the sequence of nucleic acids or the nitrogenous bases is now in the sequence of amino acids. Let's have a look at some of the ingredients and then proceed to the intricacies of translation. There are several things out of that. I think the foremost to understand is about the information itself. What information of nucleic acids can code for can code for amino acids, a sequence of amino acids. So basically, if we have a look at this, the mRNA, that is the messenger RNA, and the messenger RNA has open reading frames. And the open reading frames could be in several ways. The open reading frame starts with, say for example, the open reading frame starts with a start codon. So the mRNA has open reading frames. What are open reading frames, or ORF? They start with the start codon, that is AUG, and end with the stop codon, that is TGA. Here we're talking in terms of DNA, so we'll say it as TGA. If it were RNA, we would have called it as start codon AUG and stop codon UGA. So there could be multiple open reading frames, and you have to see all the frames on the top strand as well as on the bottom strand. If we have started from the alphabet, from the first here, from the G here, that is one reading frame, and then we will count in numbers of 3. We could also do the same with the second base like this and start counting in numbers of 3, or we could have started from the third base, and each of them could have been a codon, and so each of these are reading frames. And open reading frame always starts from 5' to the 3'. Similarly, you can also, because we said 5' to 3', you could also start looking for open reading frames starting from here, or from the first base here, second one, or from the third base. So in total we have about 6 reading frames, out of which the open reading frame, what we say as, should have a start codon. We will discuss what start codon is in a while, and it should have continuous codons like this, and it should end with a stop codon. That is an open reading frame, and they code for amino acids. The language of nucleic acids is, at least for the protein coding genes, is 3 bases. That is, one codon is the subunit. It's like the alphabet. and these several alphabets make a word. You can think of it that way. So what may happen is, as is given in the lower one here, we see an open reading frame in the lower strand, and here we have in case of B we have two open reading frames, one, and the open reading frame 2 is starting at a different open reading frame. In fact, open reading frame 1, ORF1 and ORF2 are overlapping. That could be the case. So all this information or the mRNA makes sense only in the light of the genetic code. The genetic code consists of, it is the basis of what three nucleotides or what codon codes for which specific amino acid. Say if this is the genetic code consists of three. Say for example, in the first one here we have UUU and it codes for phenylalanine, and UUC also codes for phenylalanine. Similarly, you can have UUA and UUG, which codes for leucine. We don't really have to mug up all of these codons and what codes for which amino acid, what codon codes for amino acids. But it will be important to remember the start codon, that is AUG, and the stop codons, that is UAA, UAG, and UGA. Stop codons in the sense that they do not code for any amino acid. So that is where the translation terminates. What will be interesting to know is that the genetic code is degenerate in the sense that there are multiple codons coding for the same amino acid. Say for example, we have arginine. Arginine is coded by four codons here and 2 codons here. Similarly, you may have a glycine that is coded by 4, and what we notice is that multiple amino acids are coded by multiple codons. One amino acid can be coded by multiple codons, but one codon will only code for one amino acid. That will be easy to remember that way. So there are certain rules, some of the exemptions. Genetic code is nearly universal in the sense that majority of the organisms, including prokaryote, eukaryotes, and also that of the organelles like the mitochondria and chloroplast, also obey these genetic code rules of the genetic code. Certainly there are differences, and because they have taken diverse paths, there will be subtle differences in the way this genetic code is used. So some of the times you may have GUG and UUG as start codons as well, but AUG is the start codon in all of the eukaryotic nuclear protein coding genes. And we also have some things that we need to remember about the genetic code is the codons should always be read from the 5' to 3' in direction. The second rule is that the codons are non-overlapping. In the sense, as we see here, we see that there is a codon and the next codon starts immediately after the first codon. So we do not have overlappings of the codons, or we should not see it that way. And the third simple rule is the translation is fixed. It's translated in a fixed reading frame, meaning that it starts with start codon and continuously read and terminates at the stop codon. There are certain alternatives there are exceptions in the sense that sometimes in some viruses we may have ribosomal shifting of the reading frame. It can go one base up or one base down, because of which the reading frame changes during the translation. Otherwise, in mutations, we have seen that any insertions or deletions in the open reading frame could cause the downstream codons to change, and that is what is meant by frame shift mutation. I hope we got a fundamental idea about the genetic code and we have sufficient information to proceed with the translation. We'll see you next time. Thank you.